All right, these notes go into your AP Bio spiral notebook. All right, chances are you have done Punnett squares before, at least hopefully. So this will be a review, but if these are terms that you've heard before, you're still going to write them down because they are important terms. And if you haven't heard them before, you're definitely going to write them down. All right, so a gene. A gene is a segment of DNA that codes for a trait. For example, eye color is a trait. So you have a gene that codes for your eye color. An allele is a different form of a gene. So eye color is a trait, but you could have different forms of eye color. You could have blue eyes or green eyes or brown eyes. So alleles are different forms of a gene. A gamete is a sex cell. Gametes form from meiosis. Meiosis is what creates gametes. Somatic cells are body cells. Mitosis creates body cells. Autosomal chromosomes are chromosomes that are not the sex chromosomes. Autosomal chromosomes are chromosomes that are not the sex chromosomes. So they are every chromosome except for our X and Y chromosome. Sex chromosomes are the X or Y chromosomes. Females have XX, males have XY. Those are our sex chromosomes, X or Y. A phenotype, oops, can't really see that, can you? A phenotype are the traits or characteristics that are observed. So phenotypes are usually what you see, that's what we said, but it doesn't have to just be what you see. So how a flower smells is its phenotype. How the fur of an animal feels is a phenotype. So phenotypes can be what you see, hear, smell, taste, touch, but they are things that are observed. So phenotypes are the traits / characteristics that are observed. Genotypes are the genes. They are represented by letters. So genotypes represent genes, and they are represented by letters. So heterozygous genotypes would be genotypes that have two different letters, capital and lowercase. So this is a heterozygous genotype, heterozygous, heterozygous. Heterozygous genotypes typically are hybrids as well. Whenever you're writing genotypes, whenever you're writing these letters, you need to make sure that we can tell your capital and lowercase letters apart. So I like to use cursive letters for my lowercase. So cursive S. I know cursive isn't something you guys do anymore, so you have to find some way that we can tell your lowercase letter apart from your capital letter. So even making a big P and a little p, but putting the lines where the big P is standing on it and the little p is laying on it, then that helps to tell which one's your capital and lowercase. Homozygous genotypes, remember homo is indicating the same. There's either going to be two capital letters or two lowercase letters. So this is a homozygous dominant genotype. This is a homozygous recessive genotype. Dominant alleles are represented by capital letters, and this is the allele that's expressed when it's present. Dominant alleles dominate. If the dominant allele is there, it's going to show that characteristic. Dominant alleles are represented by capital letters. It's the allele that is expressed when it is present. Recessive alleles are represented by lowercase letters, and it's the allele that is only expressed if no dominant allele is present. So you have to have homozygous recessive genotype in order to see a recessive characteristic. So recessive alleles are represented by lowercase letters. It's the allele that is only expressed if no dominant allele is present. All right, so how do we solve a Punnett square cross? Capital B is the dominant characteristic, and it represents no unibrow. Lowercase b is a recessive characteristic, and it represents an individual with a unibrow. All right, when I draw a Punnett square, I draw a square. Then I start on the top outside of the square and draw straight down, and I start to the left outside the square and I draw straight across. It really doesn't matter. I've seen different people do it a whole bunch of different ways. We just need to have 1, 2, 3, 4 spaces on the inside and 1, 2, 3, 4 spaces on the outside. When you're using a Punnett square, the spaces on the inside represent the genotypes of the offspring. The spaces on the outside represent the alleles of the parents. So there'll be 2 letters on the inside of each Punnett square, that's a 2, and there's going to be 1 letter in each spot on the outside. So let's say, okay, I crossed capital B, lowercase b, crossed with capital B, capital B. So these are my parents. Both of them do not have a unibrow because capital B is a dominant characteristic. Draw my Punnett square. One parent goes on one side. Parents go on the outside, offspring go on the middle. Each outside spot only has 1 letter. So if I'm going to put this parent on the top, I'm going to put 1 letter in one spot, the other letter in the other spot. So if this parent goes on the top, then this parent has to go on the side. I'm going to put 1 letter in this spot and 1 letter in this spot. Okay. So there's a lot going on here, I know that. Here's my cross, capital B, lowercase b, crossed with capital B, capital B. So this parent, I actually put it on the side here instead of the top, and that's fine. It doesn't matter where you put it, so long as both letters from a parent go in the same on the same side, and 1 letter goes in each spot. So this parent I put on the top, and so here's this B and this B, green here, green there. When you fill in the Punnett square, everything that's on the left-hand side goes into each box to the right. So this capital B went into here, and it went into here. This lowercase b went into here, and it went into here. This green capital B on the top went into this square and this square. This green capital B on the top went into this one and into this one. On the inside, each spot on the inside has 2 letters and represents the offspring. Spots on the outside only have 1 letter and represents the alleles for the parents. In order to find probability, you take the number of desired individuals and divide it by the number of total individuals times 100. So if I want to find the probability of having an individual with a unibrow, first I need to look to see how many of these are going to have a unibrow. Capital B is no unibrow, and capital B is dominant. So if it's there, then that's what we'll see. Dominant alleles, so no unibrow. Dominant alleles, so no unibrow. Here is a dominant and a recessive, but since I have a dominant, that's going to dominate, and I'm not going to see that characteristic. So this will be no unibrow, no unibrow. So in this situation, the probability of a child having a unibrow from those 2 parents, there's 0 that have the desired characteristic divided by 4 total offspring, which gives me 0%. All right, so let's say that we cross 2 different parents. This is a parent with no unibrow. Focus for me. There we go. I'm going to cross 2 parents, and neither of them have a unibrow. So this is a parent with no unibrow crossed with this is a parent with no unibrow. So I'm going to take 1 parent, and I'm going to put it on the top. And I'm going to take the other parent, and I'm going to put it on the side. So capital B, lowercase b, capital B, lowercase b. Everything that's on the left gets written into each box to the right. So this goes in here and here. This goes in here and here. Everything on the top gets written into each box below it. So this goes in here and here. This goes in here and here. So if I want to know what's the probability of a child with a unibrow for this pairing, capital B, both are dominant, both are no unibrow, so no unibrow. Dominant allele will overshadow the recessive, so no unibrow. Dominant allele will overshadow the recessive, so no unibrow. This is recessive, so unibrow, so this individual will have a unibrow. So I have 1 child with a desired characteristic divided by 4 total offspring, which gives me 25%. All of these represent examples of autosomal inheritance. Autosomal inheritance are characteristics that are carried on the autosomes. And remember, autosomes are chromosomes that are not the sex chromosomes. So these are autosomal inheritance are when the characteristics are carried on the autosomes. The X and Y chromosomes, however, they do have DNA, so they do have genes, so it is possible to have sex-linked inheritance. Sex-linked inheritance are when we have characteristics that are carried on the sex chromosomes. Remember, sex chromosomes are the X and Y chromosomes. Female is XX, male is XY. So sex-linked inheritance is when the characteristics are carried on the sex chromosomes. Some things that you need to keep in mind when you are looking at sex-linked inheritance: male/female matters. So male and female matters. So we have to make sure that we take that into account as well. So we have to use X and Y and the letters for the alleles. So in sex-linked inheritance, male and female matters. So when we're doing our Punnett squares, we have to use X and Y and letters for the alleles. The most common sex-linked traits are carried on the X chromosome, and that means that males are more likely to have the trait. Males only have one X chromosome, so they don't have a second X chromosome to dominate or to mask the characteristic. Most of the common sex-linked traits are carried on the X chromosome. That means that males are more likely to have the trait. They only have one X chromosome, and so no second chromosome to dominate or mask. So we're going to do an example, and this example is going to involve the sex-linked trait of hemophilia. So hemophilia is an X-linked recessive trait. So capital H is regular blood clotting, lowercase h is hemophilia. So here are our parents. XX is female. This female is heterozygous for the trait of hemophilia. Capital H, lowercase h. XY is male. This male has one capital H because it's carried on the X chromosome, so he doesn't have a sex chromosome, so he only has one allele for hemophilia, and he has the dominant allele, so he's a male with no hemophilia. All right, so we set up our Punnett square. I put the male on top here, so one allele goes into each spot. Put the female on the side, so one allele goes into each spot. Everything that's on the left gets written into each box to the right. Everything on the left gets written into each box on the right. Everything on the top gets written into each box below. Everything on the top gets written into each box below. So this is XX, so it's a female. Two dominant alleles, so no hemophilia. XX, so it's a female. We have one dominant and one recessive, but the dominant is going to dominate and mask the recessive, so no hemophilia. XY is male. We only have one allele for hemophilia, so since it's a capital H, no hemophilia. XY is male. We only have one allele for hemophilia, so since it's a lowercase h, this child has hemophilia. When you are doing probability for sex-linked traits, you have to be careful and watch what are they asking for specifically. Are they asking for how many males have hemophilia? Are they asking for how many children have hemophilia? It makes a difference. So if we're saying how many children have hemophilia, I have one child, or what's the probability of a child having hemophilia? I have one child with the desired trait, one child that has the trait out of 4 total children. So it is 25% chance of a child of these parents to have hemophilia. However, if we want to know based on these two parents, what's the probability of a boy having hemophilia, there is one male with hemophilia out of 2 males total in the Punnett square, which is a 50% chance of a male with hemophilia. So watch the question and make sure you pay attention to the wording and that you're answering what they're asking. And finally, not all inheritance is straightforward. We could have incomplete dominance. Incomplete dominance is where neither characteristic dominates. Heterozygous individual is an intermediate between both. So capital R is red, lowercase r is blue, but capital R isn't dominant and lowercase r isn't recessive. So both of them have an intermediate between them it's capital R, lowercase r, it's going to be a purple color. So neither one of those dominate, and so they're both shown at the same time. Codominance is another pattern of inheritance. Both alleles are exhibited if present. So if you have a blood type A, B, we have different blood types. We have blood type A, blood type B, blood type O. But once again, there's 2 letters for a genotype. So you can be AA, or you can be AO, or you can be AB, you can be BB, you can be BO, you can be AB, or you can be OO. So these different blood types have a different type of dominance for them. So A and B are codominant to each other. O is recessive to both of those. So if you're AO, you're going to be A blood type. If you're AA, you're going to be A blood type. If you're AB, you are AB blood type. BB, you're B type. BO, you're B type. AB, you're AB. The only time you're ever type O is when you have 2 O alleles. Fur patterns also have a tendency to show some codominance as well.